Okay, so we have the stages of mitosis. Mitosis occurs after G2 of interphase. Can anyone tell me what happens in G2 of interphase?
There's the second gap stage of interphase, and also, like things are, I forget.
It's like another time of growth in the cell and like more synthesis of new molecules and stuff.
Correct. Okay, so the first stage, prophase. So in prophase, chromatin fibers, they become more tightly coiled and they are made into chromosomes. The nucleoli disappear, and each duplicated chromosome appears as two identical sister chromatids joined at centromeres, and the centromeres just join them together in the middle. Also, the mitotic spindles form, composed of centrosomes and microtubules. Can anyone tell me what the mitotic spindles are? We kind of went over it in the last PowerPoint. Kate?
It's like the formation of all the microtubules that start to like pull on different parts of the cell.
Yeah, exactly. And then the centrosomes start to move away from each other, and the microtubules are lengthened between them. So on the blank side, we're going to draw prophase. Okay. So we'll start off with the cell. Okay. Then we'll draw the centrioles. They start to move away from each other. These are the centrioles. Oh, yeah, do it in blue. I'll do blue.
Wait, no, it's not blue.
Yeah,
Do pink.
Yellow.
Do pink.
Yellow.
Pink?
Yeah.
Pink, okay, pink.
Democratic here.
Are you pink?
Okay, so then they start to move away from each other, and then the microtubules are formed in between them. And then in the center, the nucleoli starts to disappear, and that's just representing it like going away. Yeah. And then in the middle, you have the you have the chromosomes. Okay, does everyone get it? This is prophase.
So you're saying that the chromosomes are no longer confined by the nuclear envelope?
Yeah, exactly. Oh, and then in the middle, you can draw the chromatin. Or the, yeah. Okay, so does anyone have any questions on that?
What do the red dots represent?
They're the centromeres.
Okay.
Wait, so the mitotic spindles, are those the lines in between?
Yeah, the
two
lines in between
centrioles?
that are formed.
Okay, and the chromosomes aren't lined up in the middle of them yet?
No, not yet. They're just not confined by the nucleus anymore.
Okay, and this is prophase?
Yes.
And the nucleus is like basically just, it just
Yeah, it's
dissipates.
disappearing, yeah.
Okay.
Okay, so after prophase, the next stage is prometaphase, and now the nuclear envelope is completely disappeared. The microtubules now go through the center of the nucleus in the cell, and now each chromosome has a kinnicature at the centromere. And can anyone tell me what the kinnicature is? We went over it with Sophia. Yeah.
It like holds together the chromosome?
Yeah. And it aids in like separating it. Okay. So the some of the microtubules attach the kinnicatures, and that helps pull them apart. And those are called kinnicature microtubules. And the microtubules that do not connect to the kinnicatures connect to like opposite sides of each other and aid in pulling apart the cell. I'll explain it like when we draw it. Okay, so we have the cell again. rest of the code. Okay, and now the microtubules are now through the nucleus, or like the center of the cell. And then you have the chromosome. You have the chromosomes in the middle. And then some of the microtubules are connected to the kinikatures of the chromosomes to aid in like pulling them apart for separation. And then the ones that aren't attached interact with the opposite side of the cell.
Okay, so the third phase of mitosis is metaphase, and this is the longest stage, and it lasts about 20 minutes. In the metaphase, the centrosomes have now moved to opposite sides of the cell. So you see that here, they're both on separate sides. And then the chromosomes meet in the middle, and they lie on the metaphase plate, which is just like an imaginary like boundary in the middle that they all meet on and line up. Then each chromosome, the kinicature of the sister chromatids are attached to the kinicature microtubules coming from the opposite poles. So like here it's just showing you that like this one comes here and connects to the middle, and then like but some of them also connect to like the opposite poles. They don't connect to the chromosomes and the kinicature.
What's the difference between the connecture and like a sister chromatid cohesion? Like, is it just because they happen in different stages?
Wow. The sister chromatids are like right here. Like this is what like they attach to, and then like they each come from each side and attach to one side of it. So then like later on in the phase when they pull it apart, like this like one will pull like half that way and this one will pull half that way.
Okay.
Is that no? Yes, no. Okay.
So is it like the kinikatures are like where the microtubules like attach to the sister chromatids on the outside, and then like the inside is like where the two sister chromatids like are fused together?
So
Exactly.
that's the cohesion where they're on the inside.
Exactly.
Yeah. Can you repeat that?
So like the outside is like where is the kinikatures, and like that's where the microtubules attach. And then the inside is like where the two sister chromatids attach to each other.
Okay. Exactly.
So it's just like each location.
Good handle on that.
Any more questions? No? Okay. Alright, and then anaphase is the next phase, and this is the shortest phase. This is when the two chromatids separate, and then the two chromosomes become two separate chromosomes. So instead of being like one full one, like in the here, they become two, and they pull apart to opposite poles of the cell. Yeah. This also like the cell in the anaphase starts to get longer, and so it elongates the non-kinicature tubules. So like these lines that go across all the way, they get longer, and then these ones that are attached to the sister chromatid get shorter as it pulls closer to the opposite poles. Does everyone understand that? Okay. And then by the end, you have two like equal sides of chromosomes on each side. Yes.
Does the cell copy like all the other organelles too? And do those split at the same time, or is this just like?
Not right now.
So like when the cells split, they don't all have mitochondria and stuff?
They do. It doesn't happen right now.
Oh, okay. Oh, But when, right after they split, they're just cells with
Well,
DNA.
Yeah, they're gonna split, and then in the next phase, the telophase, is when the nucleus starts to form and everything.
So cytokinesis really beginning in anaphase,
Yes.
this movement of
Towards
the
the end.
cellular stuff.
I just don't get how a cell can exist for any amount of time without having like all the like.
Oh, it does have all the organelles.
Okay.
It does.
This is just
Okay.
showing how like the
Are they just not
chromosomes
shown, or-
separate.
Yeah, they're just not shown.
Does that happen in interphase, like during the growth stage?
Yeah, you have to understand, the organelles are always there. It's just, she's not showing them because, you know, at this,
I know, but like, let's say that a cell starts out with one mitochondria and now it's splitting.
oh yeah, it
Is
has to
mitochondria-
duplicate.
Okay.
That's kind of stuff that's been happening in G2, where the cells growing all
Yeah,
these.
but the cells exist without the both mitochondria.
No, it needs
It's
the
still one
mitochondria.
cell right now.
Yeah, that's still one cell.
There's only needs one.
It's just copying.
And in
So, like-
G2 is where it has copied all that
Yeah.
stuff.
Oh,
all,
so
like
that's all
mitochondria
copied
things.
before this
Yeah,
stuff starts.
And even cell membranes can be made and stored as empty vesicles
Yeah,
so
okay.
that the cell can split.
But that stuff happens before this. Before
Right.
Okay. Not after.
I don't know, doesn't the cell like already have like the genetic material to be able to make any of that if it's lacking?
Yeah, yeah.
Because that's what it's doing with the centrosomes on both sides.
And so this is just still like, it's still just one cell and they're just separating each other. And so
Yeah,
it still has
I was
like,
just
all of
wondering
its
because
stuff.
like at some point they were gonna have to split, and I was wondering if they had this stuff at that time.
We'll get to that. Okay, so after the chromosomes split and are equal on each side, then we move into telophase. And telophase is where the two daughter nuclei begin to form, so they start making two separate nuclei, so that when the cell splits, each one will have one. And then the nuclear envelope becomes part of the cell. And you can see this here. It's like, here's the nucleus forming, and then the nucleus envelope around it forms, and then a nuclear envelope is the membrane in the eukaryotic that encloses the nucleus, so it separates it from the cytoplasm. So like, this is like gonna be like a membrane here, and then it's gonna be separated from the cytoplasm around it. Now during telophase, the chromosomes become less dense, and they are like pushed off to both ends. And then here is when this cell is like ready to divide and separate into two separate cells, each having their own nucleus, which is kind of what mitosis is. Like, that's what it is. It's like the separation of two cells, or the separation of one cell into two. Okay. Questions? No? Okay. And like cytokinesis is kind of like going along at the same time. Cytokinesis is like kind of like when it forms this cleavage furrow here. Do you have a furrow? Right there. This is what pinches the cell into two, so it like starts to, here you can see how it's like kind of like curving in, so it's starting to pinch it to form two separate cells. And that is called the contractile ring, which is made up of actin and microfilaments associated with the molecules, and the myosin. So the actin and the myosin are also the same proteins that are used in muscle contraction. So in the cell, it starts to like contract the ring, and it starts to like pull it apart, and then this decodes it into the cell until it is completely separated into two. Yeah. Everyone understand? Yeah. Oh, okay. All right. And that is all for like, the whole mitosis thing is for eukaryotic cells. In prokaryotic cells, they use a process called binary fission, which is like for their cell reproduction because they don't have a nucleus. And so first the cell grows to a maximum size, so it grows bigger. While that happens, the DNA is copied exactly. So you've got that part so far. So it gets bigger and the DNA is copied, and then they start to form a cell wall in the plasma to begin to divide it in. And okay, and And the new cell wall that they form is called the septum, right here. And then this is just like it begins to grow across the center of the cell here, being able to form two completely separate cells. And some prokaryotes, like, they don't have this, and they use the cleavage furrow that pinches the cell into half, but most prokaryotes just use the septum, which is like the cell wall that divides it in half. And after it is completely formed, the cell wall, the cells are just separated into two. So then that's how they're two separate cells.
You know, another important thing to note too is that with binary fission, the cells, when they're preparing to divide, they're also in the process of dividing at that same time. So by the time they're done preparing, they've split. And this allows them to grow much faster than eukaryotic cells. Eukaryotic cells have a lengthy preparation, right? Interphase.
Yeah, this happens much quicker than mitosis and all that.
So what are they preparing if they're already divided?
I don't understand the question.
Like, you said that while they're preparing, they're dividing, but what are they preparing if
They're
one thing is
preparing, like they're replicating the chromosome while the cell's in the process of dividing. They don't prepare everything first and then divide. Like our eukaryotic cells, they prepare everything first. G1, S, G2, then they divide, which is prophase, metaphase, interphase, telophase.
Is it more room for error then, or is it no?
It's not so much that, it's the fact that they have no, like she was saying, there was no nuclear envelope there. So there's no, it's not a true nucleus. And bacteria have only one chromosome and it's circular. We have linear paired chromosomes.
So what would happen if something wasn't?
Oh, mutations happen all the time in bacteria.
Okay.
Is that why it's so easy for them to mutate, because they don't have like a set process, it's just
Yeah.
happens so fast?
Yeah. There's other reasons too, but we're not gonna get into that today. That's stuff that we do cover later on.
Yeah, so like he's like saying like the wall like starts to form before like as it's like not it's like separating and the cell wall in the middle is starting to form all at the same time. So that's why it happens a lot faster. They don't have to the metaphase was 20 minutes long just for like all of that to happen, and this happens in just a couple minutes.
Okay. We still have other slides to present.